somewhere near the Arctic, where the wind cuts like ice, and even the ground hardens like stone. A scientist bends down toward a gray-white fragment on the ground. It looks like a rock, but it isn't. This is the tooth of an animal that lived millions of years ago, defying time. How much time has passed? Around 24 million years. And inside this tooth, something unexpected is hidden, the oldest proteins on Earth that we have been able to read so far. In this video, we will tell the story of how microscopic protein fragments from the tooth of a rhino that lived 24 million years ago have launched a new era in biology. Why are proteins so important? Why is 24 million years a record? How could this discovery change the tree of life and our ability to see the past? And in the end, how radically do these few molecules inside a single tooth show us what we might be able to do in the future? First, let's rewind the scene. We go back 24 million years. The Earth is different. The high Arctic regions that are now part of today's Canada were not as icy and harsh as they are now. The climate was milder, the vegetation completely different. In this ancient landscape, a relative of the rhinoceros is roaming, different from the African rhinos we know today, but a distant member of the same lineage. It goes down to the water, grazes, moves with its herd. Then one day, just like millions of other living beings, it falls, is buried, and its bones and teeth, within the sediments, slowly turn into fossils. Most of the skeleton is lost over time, but the teeth are stubborn, especially the inside of the tooth enamel, one of the hardest tissues in nature, almost like a molecular safe. And this is exactly where our story begins today. The tooth of a rhino ends up in the hands of scientists 24 million years later, in a freezing landscape. In recent years, you've been hearing ancient DNA stories a lot. The genome of Neanderthals has been decoded, mammoth DNA has been read, the genetic codes of animals emerging from beneath the ice have been analyzed. But there is a critical limit here. DNA is fragile, especially in warm climates. Over millions of years, it breaks into pieces and becomes unreadable. Most of the ancient DNA samples that have remained intact until today are generally on the scale of a few 100,000 to a few million years, and most of them come from cold or frozen environments. This is where another player steps in: proteins. Proteins are the molecules that DNA instructs. They are the real workers that carry structure and function in bone, tooth, and tissues. Inside some tissues, especially tooth enamel, they can survive much longer than DNA. For the last 10 to 15 years, the scientific world has been moving into a new field called paleoproteomics, the art of reading and comparing ancient proteins inside fossils. Previously, there was a general assumption like proteins can last at most 3 to 4 million years. Then what happened? A single tooth shattered that limit. Think of tooth enamel as the steel armor of the body. Extremely dense, almost no blood vessels, minimal contact with the environment. Molecules trapped inside it are held as if they were steel embedded in concrete. New studies are using exactly this property. Scientists take protein fragments at the nanogram level from fossilized tooth enamel, separate them into amino acid sequences using high-precision mass spectrometers, then compare these sequences with the proteins of living animals. In a sense, they are asking this question: the owner of this tooth, which of today's species was it more closely related to? So now we no longer just say this is a rhino, we can ask, which branch of the rhino family is it closer to? Here is the interesting part of the story. This record doesn't only come from the cold north. A research team examines fossil teeth from the Turkana Basin in Kenya, Africa. This is one of those regions that crack from the heat, which means the harshest environment for molecules. These fossils include ancient rhino species, elephant relatives, mammals between 10 to 18 million years old. And they see this. In the youngest teeth, Surprisingly, intact protein sequences are still present. Even this, by itself, is striking, because it shows that, if you find the right tissue in the right place, you have a chance to look tens of millions of years back, even in warm climates. But the real break comes with the tooth from the Arctic. The second study focuses on a fossilized rhino tooth found in northern Canada, near Devon Island, dating around 21 to 24 million years ago. What comes out of this tooth? Not a full genome, not even complete protein sequences, but over 1,000 short amino acid chains belonging to 7 different tooth enamel proteins. In other words, small, fragmented, but still readable remnants of the proteins that once formed this animal's tooth enamel. Even these tiny fragments are enough to reinterpret which rhino lineage the animal was closer to, and where, on the tree of life, its branch actually sits. And they allow us to say, the oldest animal proteins ever read to date have officially been found. This discovery pushes the scientific shelf life of ancient proteins back by about 14 to 15 million years. So, what do these molecular crumbs give us? By looking at a fossil bone, we could already say, this is a rhino. What do proteins add? This. Small differences in the protein sequences in tooth enamel act like signatures between species and lineages. Just as mutations in DNA help us build family trees, changes in protein sequences can also show who is related to whom and how closely. Thanks to this, we can test whether estimated relationships based solely on bone shape are right or wrong. Ask again, did this species really belong to this family, or was it closer to a different branch? That is exactly what happens in the rhino example. Ancient tooth proteins reveal unexpected connections in the rhino evolutionary tree and require some branches to be rearranged. The work that we could previously do only up to 4 million years back, we can now push beyond 20 million years. Now, we come to the truly critical part. Is this just a rhino story? No. Do you know what this technology means? Teeth from early ape and human ancestors related to human evolution could become readable at the protein level even in the warm African climate. Even at ages where we say DNA won't survive, we have a chance to find molecular clues about which species is the ancestor of which. We could see much more clearly how mammal lineages branched and diversified, how they responded to climate changes, and which species entered the stage and which ones disappeared at what time. And one more question, if tooth enamel preserves this well, what other tissues might be carrying molecular archives we haven't discovered yet? This discovery is not just a record-broken headline, it gives scientists this message. What you have thought impossible for years might simply be because you were looking at the wrong tissue. Here's the question that immediately comes to mind. So then, can we get dinosaur proteins too? Will Jurassic Park become real? Scientists are cautious here. Dinosaurs are 65 plus million years back. Even 24 million-year-old proteins could only be found in fragmented form under very special conditions, in an ultra-durable structure like tooth enamel. There have been previous studies suggesting there may be protein remnants in dinosaur fossils, but most of them are controversial and have not been able to deliver a clear paleoproteomic picture. So, reading complete dinosaur protein sequences with high confidence is still far beyond the current limits. But for mammals, birds, and all kinds of animals in the 10 to 30 million year range, the window is opening wider and wider. This is not Jurassic Park, but it does bring us this: the chance to fill dozens of pages of the book of evolution that we've assumed were completely dark until now, with molecular data. Sometimes in science, knowing what we cannot do is just as important as knowing what we can do.